Welcome to the Biological Blueprint, where we'll decode the 6 fundamental bases of animal classification. This video will explore the architectural framework that structures the immense diversity of the animal kingdom.
The immense diversity of the animal kingdom is categorized using 6 foundational bases of biological architecture, or the 6 pillars of classification. These pillars include the levels of organization, body symmetry, embryonic layers, the coelom, body cavity, segmentation, and the notochord.
Pillar 1 is the levels of organization, ranging from cellular, like sponges, tissue in Coelenterata and Ctenophora, only organ level in Platyhelminthes, to organ system, found in Aschelminthes to Chordata. As complexity increases, cells group into tissues, then organs, and finally, organ systems.
Variations in system complexity are visible in digestive systems, which can be incomplete with one opening or complete with 2 distinct openings. Circulatory systems start from annelida and can be open or closed, with blood circulating through sealed vessels in closed systems.
Pillar 2, architectural symmetry, defines how an animal's body can be divided. Asymmetrical bodies, like sponges, have no plane of division. Radial symmetry, seen in coelenters, ctenophores, and adult echinoderms, divides the body into identical halves along the central axis. Bilateral symmetry, from Platyhelminthes to Chordata, is divided into identical left and right halves by one specific plane.
Pillar 3 concerns embryonic germinal layers. Diploblastic animals like coelenterids have 2 layers, ectoderm and endoderm, separated by an undifferentiated mesoglia. Triploblastic animals, from Platyhelminthes to chordates, introduce a 3rd layer, the mesoderm.
Pillar 4 defines the coelom, or body cavity. Acoelomate animals, such as Platyhelminths, have no cavity. Pseudocoelomate animals, like Aschelminths, have a false cavity with scattered mesoderm pouches. Coelomate animals, including annelids to chordates, have a true cavity fully lined by mesoderm tissue. Pillar 5 is metameric segmentation, where the body is externally and internally divided into repeating segments. This modular blueprint is classic in earthworms, Annelida, and is also seen in Arthropoda, external, and Chordata, internal.
Pillar 6 is the notochord, a mesodermally derived rod-like structure providing dorsal structural support during embryonic development. Chordates possess this rod, serving as the foundational axis, while non-chordates, peripheral to echinoderms, never form this structure.
Synthesizing these pillars creates a master taxonomic tree of the animal kingdom. By applying the levels of organization, symmetry, and coelom types sequentially, we can map the precise classification of each phylum.
This dashboard summarizes the basis of classification. It illustrates the progression from cellular to organ system organization, the 3 types of symmetry, diploblastic versus triploblastic germ layers, and the 3 coelom types.
Next, we delve into the non-chordates, a systematic guide to invertebrate life. This section will classify the first 10 phyla of the animal kingdom based on the evolutionary blueprint we just discussed.
Phylum Porifera, the sponges, are the most primitive multicellular level, exhibiting mostly asymmetrical body, except Cycon, which is radially symmetrical and echoelomate status. They lack organ systems and rely on a water canal system for essential functions like digestion, respiration, excretion, and reproduction, using specialized cells like choanocytes and amoebocytes. They are bisexual/hermaphrodite, showing both sexual and asexual reproduction with indirect development.
Phylum Coelenterata, or Cnidaria, are the stingers, with a tissue-level, diploblastic, and acoelomate organization, exhibiting radial symmetry. They exist as polyp, sessile, asexual, or medusa, free-swimming, sexual forms, with life cycles showing metagenesis. Their key feature is cnidoblasts for defense, offense, and prey capture.
Phylums Caetanoptera, the comb jellies, possess tissue-level organization, radial symmetry, and are diploblastic and acoelomate. Distinctively, they use 8 rows of ciliated comb plates for locomotion and exhibit bioluminescence, using sticky colloblasts to catch prey.
Phylum Platyhelminthes, the flatworms, are triploblastic, bilaterally symmetrical, and acoelomate. They show an organ level of organization with an incomplete digestive gut. Many are parasitic, utilizing specialized hooks and suckers, and some, like Planaria, have high regeneration capacity.
Phylum Aschelminthes, the roundworms, represent the first to achieve an organ system level and a pseudocoelomate body cavity. They are triploblastic and bilaterally symmetrical. Their digestion is complete, and they exhibit sexual dimorphism, with females typically longer than males.
Phylum Annelida, the segmented worms, are coelomates with organ system organization, bilateral symmetry, and metameric segmentation. They possess a closed circulatory system and specialized nephridia for excretion. Notable specimens include the aquatic nereis, earthworms, and blood-sucking leeches.
Phylum Arthropoda, the jointed appendages, is the largest phylum, characterized by jointed appendages and an external skeleton. They feature an open circulatory system, complex sensory organs, and specialized systems for respiration and excretion. This group includes crustaceans, arachnids, myriapods, and insects.
Phylum Mollusca, the soft-bodied, are organ system level, coelomate, and bilaterally symmetrical. They typically possess a shell, a muscular foot for movement, and a mantle covering the visceral mass. They also utilize a rasping organ called the radula for feeding.
Phylum Echinodermata, the spiny-skinned, are unique as adults show radial symmetry, despite being bilaterally symmetrical as larvae. They have an organ system level and are coelomates. Their distinctive feature is the water vascular system for locomotion, capturing food, and respiration.
Phylum Hemichordata, previously considered a subphylum of Chordata, are now classified as non-chordates, as they have a structure similar to notochord, i.e. stomochord, ectodermal origin. They possess organ system level organization, bilateral symmetry, and a coelomate body plan. Their body is divided into proboscis, collar, and trunk, and they have an open circulatory system.
This completes our systematic overview of the non-chordate phyla. This diagnostic matrix traces the structural evolution across 10 non-chordate phyla. We can see the progression in the level of organization, symmetry, coelom, and segmentation as we move through the phyla, starting with Porifera and ending with Hemichordata.
This second matrix focuses on physiological complexity, examining the digestive, circulatory, and respiratory systems. Note the gradual development of complete digestive systems and the later appearance of circulatory and respiratory systems, alongside distinctive features for each phylum.
Before moving to phylum Chordata, here are some salient features that differentiate Chordata and non-Chordata from each other. The most important one is their mesodermally originated notochord.
The Universal Chordate Blueprint details the 4 fundamental traits present in all chordates during their embryonic stage. These are the notochord, single, dorsal, and hollow nerve cord, paired gill slits, and the postanal tail. Phylum Chordata is mapped into 2 groups, Acraniata/Protochordata and Craniata/Euchordata. Protochordates include subphylum Urochordata and Cephalochordata, which are exclusively marine, whereas Craniata includes subphylum Vertebrata, having jawed, gnathostome, and jawless, agnathostone animals.
Urochordata, or Tunicata, have a notochord only in the larval tail. Examples include Ascidia. Cephalochordata, like Branchiostoma, have a notochord extending from head to tail that persists throughout their life.
Subphylum Vertebrae is further divided into division Agnatha, jawless, like Cyclostomata, and division Gnathostomata, jawed vertebrates. Gnathostomata further divides based on the presence of fins into superclass Pisces and limbs into superclass Tetrapoda. Superclass Pisces includes class Chondrichthyes and class Osteichthyes, whereas superclass Tetrapoda includes class Amphibia, Reptilia, Aves, and mammals.
Class Cyclostomata includes the jawless ectoparasites, like Petromyzon, lamprey, and myxine, hagfish. They are marine but spawn in fresh water, have a closed circulatory system, and a cartilaginous endoskeleton. They lack scales and paired fins, but have 6 to 15 pairs of gill slits.
Now we move on to gnathostomes. Class Chondrichthyes of superclass Pisces are the cartilaginous predators, such as sharks and rays. They have a cartilaginous endoskeleton, a 2-chambered heart, placoid scales, and are cold-blooded. They lack an air bladder and must swim constantly to avoid sinking. They also lack operculum, gill cover, but have notochord persistent throughout the life.
Class Osteichthyes, the bony fishes, have a bony endoskeleton, cycloid or ctenoid scales, and streamlined bodies. They possess 4 pairs of gills covered by an operculum and an air bladder for buoyancy control. Most are oviparous with external fertilization.
Moving on to superclass Tetrapoda, the first one is class Amphibia, or the double life animals, inhabit both aquatic and terrestrial environments. They have a 3-chambered heart and are cold-blooded. Respiration is multimodal, using gills, lungs, and skin. Reproduction involves external fertilization and indirect development.
Class Reptilia, the creeping animals, are first true land vertebrate, mainly terrestrial with dry, scaly skin. They are ectotherms and respire only through lungs. They lay shelled eggs on land, marking a major adaptation for terrestrial life.
Class Aves, or the birds, are characterized by the presence of feathers, a beak, and forelimbs modified into wings for flight. They possess an internal air sac system for efficient respiration and are homeotherms, maintaining a constant body temperature. They don't have any gland except of uropygial or oil gland and have crop and gizzard as an additional chambers in alimentary canal. Bones are fully ossified and pneumatic.
Class Mammalia is defined by mammary glands for nourishing young and body hair. They are homeotherms and viviparous, giving birth to live young except of Ornithorhynchus, duck-billed platypus, which are oviparous. They possess a 4-chambered heart and specialized teeth heterodont, adapted to various diets. This concludes our systematic study of the phylum Chordata, tracing the evolution from primitive jawless fish to the highly developed mammals. We've explored the unifying chordate features and their diversification. Thank you for exploring the biological blueprint of the animal kingdom. The classification pillars provide a robust framework for understanding the stunning variety of life on Earth.